All right, welcome to AP Biology, and this is our cell respiration part 2 video. So a couple of things we do need to remember. Once again, the structure and function of the mitochondria, that inner folded membrane, what does it do? It increases surface area. What does that allow us to do? It allows the mitochondria, not us, but the mitochondria to have lots of electron transport chains, making it nice and efficient. Okay, the other thing we want to reiterate once again is the molecules of energy that we are consuming in terms of food. The amount of potential energy is related to the number of hydrogen atoms we do find in each molecule, which is the reason why fats are so efficient at storing energy because of the high amount of carbon-hydrogen bonds and those representing all the hydrogen molecules stored there. The hydrogen is not a form of energy, it just represents the energy in those stored. Alrighty, so let's look at our big picture. Remember, we have already talked about glycolysis, and now we are going to focus on what happens after glycolysis with the pyruvate molecule. And as you can see, this pyruvate molecule is going to be going into the mitochondria, and that will only happen if oxygen is present. Okay, so we've now gone through glycolysis. We've made the pyruvate molecule. We have to get that pyruvate molecule into the mitochondria. There is a transport protein that will move that mitochondria into there. Sometimes that transport protein, depending upon the organism, will consume some energy, and sometimes it does not. But as you can see on the left-hand side of the diagram, we have our 3-carbon pyruvate molecule coming in, and what's going to happen to that is going to be converted to a 2-carbon compound, and in that process we're going to rip off a CO2 molecule, which you can see in blue in the diagram. Okay, and that we are going to exhale that gets transported from our cells to our lungs via our blood, again using the bicarbonate ion. And then we are going to oxidize that 2-carbon compound. What does that mean? It means we're going to rip some electrons off of it. Where do those electrons go? They are going to be used to reduce NAD, as you can see in the diagram. An important diagram to know. Finally, our end product of this is going to be acetyl-CoA, which has a lot of potential energy. And let's step back. Remember, in glycolysis, we produced 2 pyruvate molecules from each glucose molecule. So you can double the amount of acetyl-CoA and the amount of NADH being produced here when you think about a single glucose molecule. All right, so we are going to continue onwards, and we're going to look again at what happens now in the citric acid cycle. And as you can see below, we are going to produce some ATP via substrate level phosphorylation. And remember, that's using enzymes to make some ATP. Not very efficient, but we can still make a fair amount of ATP, oftentimes enough for many organisms, especially bacteria, to get by on. Okay, the other product we're going to get here are going to be high energy electrons that are going to be carried to the electron transport chain via our electron carriers NADH and FADH2. Okay, so let's look at the citric acid cycle right here. And again, we see a lot going on. We do see that it is a cycle because we start off with oxaloacetate, our 4-carbon molecule at the top of the citric acid cycle there, and we are going to end up with oxaloacetate. And in that process, we're going to get rid of 2 molecules of CO2, so we're going to blow off the rest of the carbon that came in originally via pyruvate. We are going to use those high energy electrons in the acetyl-CoA molecule to reduce NAD to NADH and to reduce FAD to FADH2. Okay, as you can see right there, and we're going to produce some ATP. So once again, let's count up the number of NADHs we're going to get from pyruvate. We have 1 during the conversion of pyruvate to acetyl-CoA. We're going to get 3 in the citric acid cycle, and 1 FADH2 and 1 ADP. Okay, so here's another diagram, allowing you to look at how the carbons are being rearranged and moved around. Again, you do not need to know this diagram, the details. You do need to know what is coming in, i.e. acetyl-CoA. What is happening? We're going to reduce NAD to NADH. We are going to reduce FAD to FADH2, and we're going to produce one molecule of ATP. That's what you need to know. And in that process, we are going to regenerate oxaloacetate. So as you can see right there, again, we are going to completely oxidize pyruvate using that energy in those electrons once again to reduce NAD and FAD and make some ATP via substrate level phosphorylation. And there's our accounting. Per glucose molecule, we are going to produce 6 NADHs and 2 FADHs per glucose molecule, along with 2 ATPs, and that is not including the creation of an NADH from the conversion of pyruvate into acetyl-CoA. All right, so now we've done the citric acid cycle, and we've now produced these high-energy electron carriers in glycolysis and in the citric acid cycle and in the conversion of pyruvate to acetyl-CoA. So now we got to mine the energy that are in those electron carriers and use it to make ATP. And this is where we're going to produce the largest amount of ATP. And once again, what do we have to do? We have to create and maintain a concentration gradient of protons. Once we have that gradient, we can now use that gradient to do work. And so we're going to see active transport and we're going to see diffusion, just like we saw in photosynthesis, using the energy in those high energy electron carriers to pump protons against the concentration gradient. Now use that concentration gradient to let those protons diffuse down through ATP synthase. So let's look at the electron transport chain in oxidative phosphorylation. Do notice on the left-hand side of the graph that is the free energy relative to oxygen. So as you can see on the left-hand side, we have NADH coming in at the top of the chain with electrons at a relatively high level of energy. We have FADH2 coming in about midway, and then we have oxygen at the very end that is going to accept those depleted electrons, okay, depleted in terms of energy, and then we're going to reduce oxygen, and that's the reason why we breathe, is to allow to have that oxygen at the end of the electron transport chain, and our final product is going to be water. Okay, so again, where it's happening on the electron transport chain, where there are literally thousands of these electron transport chains on the inner membrane of the mitochondria. So what's going to happen? Okay, there's my NADH and FADH2. Notice that they come in at different places. NADH is going to come in at the very top of the chain, and it's going to go through 3 of these multi-protein complexes. And FADH is going to come in at about the middle of the chain, and it's going to go through number 3 and 4. So let me just kind of look at these. 1, 3, and 4 are all proton pumps, whereas number 2 is just an electron carrier acceptor. Okay, so it doesn't pump protons, but 1, 3, and 4 do pump protons. And so NADH, when it coming in at the top, will go through 3 of these proton pumps, therefore pumping 3 protons. FADH will only pump 2 protons. Okay, so again, what is the electron transport chain? Again, it's a series of proteins where each preceding protein complex is less electronegative than the succeeding one. So as the electrons are transferred from one protein to another, the protein is actually, since the the proteins are more electronegative, they're going to pull those electrons down that chain. Okay, so they're going to oxidize the uphill neighbor and reduce the downhill neighbor. And again, how does this happen? Again, each protein is more electronegative than its uphill neighbor. And what else is going to happen? We are going to reduce oxygen to make water. Okay, and in this whole process, remember, those are proton pumps, 1, 3, and 4, and we are going to be creating and maintaining that proton gradient. So once we have that proton gradient, just like we saw in photosynthesis, we are going to use it to allow those protons to diffuse down through ATP synthase. And so we're taking that potential energy of the proton gradient and turning it into kinetic energy as they move through ATP synthase, causing it to spin and causing the anabolic building of ATP from the precursors of ADP and that phosphate. And there's our picture of the electron transport chain of right there. And as we can see the NADH coming in, going through 3 of those pumps. We can see FADH only going through 2 of those pumps. And do know that, once again, ATP synthase is not part of the electron transport chain. It is separate. And again, what is chemiosmosis? It is an energy coupling mechanism that uses the energy stored in the form of a proton gradient across a membrane to drive cellular work. What is that work? It's making ATP. And once we have the ATP, your cells can now do all sorts of other complex and not so complex things, such as transporting molecules across a gradient through active transport. They can be used to drive reactions such as that are endergonic, or ones that require energy, such as all your anabolic or building reactions, and also for moving things around through motor proteins, which physically drag large transport vesicles around the cell. Okay, so this is an important diagram, and it's one you really need to know as an overview of cellular respiration. If you can draw this from memory, you're going to have a lot of the details that you need for this particular course, and just kind of knowing some of the finer points of what is happening there. But once again, just look at it. We start with glycolysis happening outside of the mitochondria. We are going to produce some ATP via substrate level phosphorylation, but more importantly, some of the NADHs, 2 of those. And then we also produce 2 pyruvate molecules, which, remember, still have about 75% of the potential energy of that original glucose molecule. So now we have to shuffle that pyruvate into the mitochondria, and that will only happen if oxygen is present. And we are going to convert the pyruvate into acetyl-CoA, which will then allow us to go into the citric acid cycle. Once again, we have created some NADH via that movement from pyruvate into acetyl-CoA into the mitochondria. And the citric acid cycle, once again, making some ATP via substrate level phosphorylation, but more importantly, ripping those electrons off of the acetyl-CoA and transferring those high energy electrons to NAD and FAD in order to reduce those. And finally, the oxidative phosphorylation of the electron transport chain and chemiosmosis to make most of that ATP. And as you can see, per glucose, we're going to get about 36 to 38 ATPs. Why the variation? Well, that depends upon whether or not we need to utilize energy to shuttle electrons across or to get pyruvate into the cell. So let's again look at this ATP accounting. The breakdown of glucose will have a delta G of negative 686 kilocalories per mole. And once again, that is an exothermic catabolic reaction, one that is spontaneous. To make a mole of ATP takes 7.3 kilocalories. Okay, so that is non-spontaneous. It is endothermic. It is anabolic. So we'll get that 36 to 38 moles of ATP per glucose, which comes out to about 40% efficient, again, which compares to the most efficient cars at about 25% efficiency rate. So it's pretty darn good. And again, the rest of that energy is going to be used to generate body heat and keep our bodies nice and toasty.